Hi, my name is Raekwon and I work at the Department of Oncology at the Sidney Kimmel Comprehensive Cancer Center at the Johns Hopkins School of Medicine. And the title of my project is Targeting Glutamine Metabolism and M2-Tumor-Associated Macrophages in the Tumor Microenvironment of Prostate Cancer. First, I'll explain what prostate cancer is. So, cancer in general is just how cells divide to multiply, but sometimes there can be errors in the replication of these cells, and these cells can become cancerous. When they're cancerous, they really divide at higher rates, and they also can invade other cells to make them divide at higher rates, causing these cells to form a tumor, a cancerous tumor, which can then spread to other parts of the body. And prostate cancer is particularly in the prostate. It's the most common cancer in men, and it's the second leading cause of death in men. And there are about 175,000 cases yearly, and there are more than 30,000 men that die yearly. And these tumors are not just tumor cells, there are immune cells present, but the immune cells are sort of in a frozen state and it can't really function to eliminate these cancer cells because they can't really detect them because of multiple mechanisms by which the cancer cells give these signals to sort of hijack how these immune cells function. And prostate cancer is particularly hard to treat because it commonly recurs and also it spreads to the bone if not detected at an earlier stage. So one of the immune cells that are present in the actual tumor, they're called macrophages. Macrophages make up a large portion of the tumor, which is why they're a good target, and they also originate on embryonic development and they're resident in all tissue in the body. So they're a potential good target for prostate cancer for those two reasons. And there are two subsets of macrophages. There are M1 macrophages, which really patrol and sort of target these invaders that it detects in the body to eliminate them by different mechanisms, either eliminate them directly or give signals to other immune cells to eliminate the invader. These cells are very plastic, meaning that they respond really well to different stimuli in the environment around them. They sort of in different signals that either the invaders give off or other regular cells give off. They can sort of communicate with each other to really eliminate these invaders that they detect in the body. And macrophages that are hijacked by prostate cancer or cancer are called tumor-associated macrophages, meaning that they don't function normally because they're hijacked by prostate cancer. Like I said before, they make up a large portion of the microenvironment. They're really long-lived immune cells that are resident in all tissue. And these cells particularly promote the progression of prostate cancer and also vascular formation and resistance to treatment. As you can see in the figure, these cancerous cells, these prostate cancer cells, once they transition, they can really convert these macrophages into M2 macrophages, what will give them the immunosuppressive ability in which they will really be suppressive and sort of help its progression in multiple ways, which even leads to the spreading of these cancerous cells to the bone marrow, which is a common occurrence. So glutamine metabolism and the M2 macrophages. So glutamine is a abundant amino acid essential to cell growth. And the downstream effects of glutamine on the suppressive ability of M2 macrophages is not really well understood, but we know that these M2 macrophages utilize glutamine a lot more than the other subset of macrophages. And one way that we can detect how glutamine impacts these M2 macrophages, so in the tumor, there are receptors on the surface of these M2 macrophages which bind to ligand, which really are just like chemicals that really are like the way these cells communicate with each other, and the cells sort of sense the environment they're in. And once these interactions cause chemical messages to relay a signal, which can lead to a response, which can affect the protein expression, which is basically affecting how these cells function. So my hypothesis is that we can use glutamine deprivation to revert these immunosuppressive M2 tumor-associated macrophages to anti-tumor promoting and M1 tumor-associated macrophages, allowing for anti-cancer immunity against prostate tumor cells. So the way I tested my hypothesis was I I use resting macrophages that aren't either M1 or M2, but they're not specialized, and I specialize them using a method that my mentor developed because it's very complicated for these resting macrophages to become M1 or M2 because they require certain signals in specific timing for these cells to become either M1 or M2. And it took years to really simulate how these cells are made into M1 or M2 in organisms. And this is the best method developed by my mentor. So the method I used was I took these resting macrophages and I made them into M1 or M2 using the protocol my mentor developed in the presence of glutamine and in the absence of glutamine. And I wanted to see how this changed how they function. And the way I measured how they function is sort of the proteins they produce. And the way I looked at the quantity of proteins is immunobody, which is basically taking these proteins and breaking them down into a linear form where I could detect them using antibodies. And flow cytometry really is to see how these cell surface markers, which are receptors that I mentioned before, how they sort of change and how the quantity is different in glutamine with glutamine and without glutamine. But I really didn't master the process of flow cytometry because it's really complicated. Another method we can measure protein is by looking at the mRNA transcripts using qRT-PCR. And mRNA transcripts are basically messages from a cell to produce proteins. So in a cell, in a nucleus, there's DNA, and DNA can't actually leave the nucleus, but it holds all the instructions to make proteins in a cell to really give them their function. So we can measure how many messages are made from this DNA to be made in the proteins because mRNA can leave the nucleus while DNA can't. So we measured how many of these messages are actually made for the proteins to be produced, ultimately changing the function of these cells. So here's tissue of an actual prostate cancer patient, and we found that in the first image, you can see that CD206, which are M2 macrophages, which are the immunosuppressive ones, and red dots, and then the green dots are the M1 macrophages, which are the patrolling ones, which would function to eliminate these cells, but they're not able to because of the communication between the cancerous cells and these M1 macrophages. And the third slide is just showing the nucleus of the cells present, which means that not only immune cells are present in the environment, but also just the cancerous cells are also present, and also just normal tissue found in the prostate. In the third image, it's all together, and you can see that how the formation of the prostate, there are a lot of these M2 macrophages in the actual tumor, which means they're a good target for immunotherapies and treatments. So my results show that in M2 macrophages, the cell surface markers and regulatory molecules are impacted by glutamine deprivation. And as you can see in the first figure, these bands show protein because the proteins that we look at are the same size, so we can sort of sort them in a way that we can compare the quantity of protein in the different samples. You can see with the downregulation of glutamine in the M2 macrophages, it's a fainter band, which means there's less protein, which means that glutamine deprivation causes a downregulation of CD163 protein, which is a cell surface marker that senses bacteria on M2 macrophages, which is a common marker found on M2 macrophages. And the graph next to it shows the mRNA transcript data, and the mRNA transcript data shows that it's an upregulation of mRNA that encodes CD163. And this shows that not all mRNA transcripts that are created are actually made into proteins, because there are a lot of steps in between the mRNA transcripts being actually converted into protein. And the next molecule next to it in the graph is IL-10, and IL-10 is an anti-inflammatory mediator, which means that it's really suppressive, and it sort of signals the immune system to sort of not be to not sort of attack invaders, and everything's like fine, and it contributes to the suppressive ability of M2 macrophages, and it's downregulated in the absence of glutamine, shown by the asterisk, and it's significant, which means that glutamine deprivation drastically impacts the expression of mRNA transcripts of IL-10. Glutamine deprivation affects M1 regulatory molecules but not cell surface markers. So CD86 is a primary marker of M1 macrophages, and it functions to sort of recruit an immune cell called a T cell to initiate an immune response to sort of get them to eliminate invaders that they detect. And as you can see, the first band is really a thick band, and it shows the CD86 expression in M1 macrophages with and without glutamine. You can see it's sort of a larger band, which means they are different sizes, and they're different sizes because proteins are glycosylated, which means that sugar groups are added, which these sugar groups sort of show where in the cell they belong. And as you can see, there aren't differences in CD86 expression in M1 macrophages with and without glutamine, showing that the cell surface markers are not really impacted by glutamine deprivation. Well, interferon gamma is a mediator of inflammation, which means that it sort of causes inflammation to sort of recruit immune cells. And with glutamine deprivation, we found that these cells upregulate mRNA transcripts of interferon gamma, which means it sort of increases their ability to sort of produces signals to increase their ability to initiate inflammation. As you can see, my results show that glutamine deprivation causes downregulation of a potent anti-inflammatory molecule called IL-10, and also causes downregulation of CD163 on a protein level in these M2 macrophages, which suggests that glutamine deprivation inhibits suppressive M2 macrophages from these given markers and regulatory molecules. So future work would be for us to actually find a more accurate way to deprive these macrophages in an actual organism, because there are discrepancies between how macrophages act in culture and also in an organism. So someone developed a drug called DAWN, which is a glutamine antagonist, which prevents the cells from actually utilizing glutamine. And they actually did a clinical trial of this with cancer patients, and they found that it was super toxic to patients because it would just target glutamine all throughout the body, which would inhibit growth of all cells, not just the cancerous ones. So at Hopkins, they developed a prodrug, which is an inactive version of this DAWN glutamine, which is a glutamine antagonist. And the enzyme that cleaves this drug to make it an active version is found in the tumor microenvironment. So we're going to test this prodrug in mice with a prostate cancer tumor to see how the growth of this tumor is impacted by this drug. I would like to acknowledge Dr. Nicole Rosen and the Ingenuity Project for giving me the opportunity to conduct an independent research project. I would also like to thank Dr. Jelani Zarif, my mentor, for accepting me in his lab and sort of teaching me the basic immunology protocols that I needed to actually conduct my research. I would like to thank Van Chin, Thomas Nerschel, the baby Tedros, and the Johns Hopkins School of Medicine for all their support and continued guidance throughout my whole research project. Thank you.